Professor Tom Johnson from University of Illinois.
Oh, press got it.
Yeah. I know Tom since already 15 years now, I realize. It's a long time, and actually I can tell that he actually saved me and my PhD because without him I probably wouldn't have a PhD. Because he was the only one in the entire world who did selenium isotopes. And my advisor in Germany decided, let's do selenium isotope, not knowing how difficult it can be. I have to say that. But, and it is difficult, and Tom was kind enough to allow me to go to his lab and do the selenium isotope measurements for 3 years there. Yeah, and that's actually, I can say that, that's a photo from, I think, 2010 or something. 2010 or something. And Urbana is here on the photo, me, Tom, and that was the crew by that time. So it was, we had a good fun time. That's actually, yeah, so Tom does this amazing isotope work with all these redox, especially redox sensitive elements, chromium, selenium, uranium, and especially crown border systems and experimental work. So I learned a lot about experiments with isotopes. That's about, it was an amazing time. So and he said I should keep it short because he has too many slides. So that's why. Floor is yours, Tom.
All right, we're moving the L closer. So I'll start out by just making sure, let's do a quick sound check because I want to make sure that the people in Zoom land can
That's why they
hear okay. So give us a thumbs up signal or something if you can hear okay. The owl is blinking its eyes. I think.
I got it not very well, that's why I moved it.
Yeah.
Yep, everyone can hear you. You got those
Okay.
up.
All right, then I will start. I want to thank Catherine for the kind introduction, and I want to thank you all for having me here. It's particularly exciting because as we've been saying, you know, when you have these areas that are so different, that come at problems from different directions, sometimes at the interface, there's great stuff. And I think that's what I'm seeing. Now, my talk is going to be very much on the geoscience side of things, but as I think you'll see, you know, even if you're not already talking to Catherine and Urbana about the potential here, I think you'll see that, you know, we geoscientists, we deal with these horrendously complex systems. They're not as complex as the human body, but we have to have these specialized tools so that we can see in deeper and get some process understanding. So, like Catherine says, I have many slides. I don't want to miss any part of this because it's a very rich story. So here I go. First thing I want to talk about is the sources in the environment of these three elements, and there are many. So those of you who remember the movie Erin Brockovich way back when, that was about chromium that was in the groundwater at this site in the Mojave Desert. This was a place where they had to compress natural gas, send it across the desert. Every 50 miles, they had a compressor station, and they had cooling towers that had this cocktail in the cooling water that had a lot of chromium-6. Hexavalent chromium is the word that's often used out in the environmental industry. And so that was what was behind the whole Erin Brockovich story. At the famous Hanford site, which has all sorts of radionuclide contamination, they also had chromium-6 contamination for the same reason. Here is a 1960 photograph of the site there. They had nuclear reactors that had cooling loops, and they ended up with a big chromium mess related to that. There are also many sites in all countries of the world, in all parts of the United States that I'm familiar with, that have chromium plating operations, right? So I don't see anything that's chromium plated in this room. It's not as popular as it used to be. But in this photo that I'm showing you here, this huge roller that might be used to do printing or crushing of grain or something like that is about to be coated with chromium. Chromium is a very hard coating. And so they would put it in this vat that would be about 1,000 liters. And I'm not kidding, there's no typo here. It's 200 grams per liter of this toxic oxidizer, hexavalent chromium. A little of that goes a long way. And so there are many chromium plumes related to chromium plating. But it doesn't stop there. Many of you, I think, probably have on your radar screen, there's an increased interest in geogenic contamination. I think it's more important than we realized. Of course, if you've been working on arsenic, you know this already. But when you look at geogenic contamination, it's actually present it's certainly in the United States. Ken Bellitz of the USGS is currently on a speaking tour where he's talking about the sources of contamination nationwide. A lot of it is geogenic. And so chromium, this is just California, and everywhere that you see the green, these are naturally occurring ultramafic high chromium rocks that tend to give a lot of chromium off to the water. And so you have naturally occurring contamination in these areas. You also see naturally occurring chromium in the Appalachian region as well. Let's skip to selenium then. The classic selenium problem was on the west side of the San Joaquin Valley for geological reasons that I have no time to get into. The rocks on the shales, the mud rocks that are very organic rich on the west side of the San Joaquin Valley in California. So on the upper left of the diagram, you can see the San Francisco Bay area. The San Joaquin Valley then is southeast of that. And the yellow portion there is where the selenium-rich shales outcrop. The soils that are developed on those shales are selenium-rich. When they came in and started doing agriculture, they irrigated those areas. They ended up having to dispose of saline irrigation wastewater, and that caused a big environmental impact in the region. And it affected, for example, rare migratory birds that had a number of deformities. Birds are known to be particularly susceptible to selenium poisoning, and fish are as well. But that's not the only place where we've had selenium problems. This photograph that you're looking at now is related to phosphate mines in Idaho, and the overburden rock is similar selenium-rich shales from a similar period in Earth history. So they remove this overburden for these open pit mines, and then they move it to someplace and pile it up, and then the water percolates through it and mobilizes the selenium for reasons that I'll talk about right now. But that's not all. Coal ash is high in selenium. Coal-fired power plant scrubber effluent is high in selenium. And then produced water from oil and gas operations is rich in selenium as well. So there are many sources. Now I'll skip to uranium, and I'm only going to focus on the uranium sites that are in the Colorado Plateau. There are some others, but as many of you know, the mining and milling operations in the Colorado Plateau region created a number of sites that have uranium contaminated soils. And in particular today, I'm going to focus on this site. This is the so-called Rifle site. The river that you see there, this is the upper reaches of the Colorado River in kind of central western Colorado. Interstate 70 is just south, just off of the photograph here. And those homes that you see, those are the outskirts of the town of Rifle. But inside the, let's see, I'm not sure if people are going to be able to see my cursor.
They can.
They can. Okay, question is, can I see my cursor?
Yes.
Here it is. Okay, so there we go. So in this area, there was a uranium milling operation, and the Department of Energy trucked off many truckloads of soil that was uranium contaminated, but they still had a problem with the groundwater. So these contaminants tend to be persistent. It's hard to really get rid of them for geologic reasons that I won't get into. And still, there was, and probably still is today, considerable uranium that is moving from this area where the uranium mill was, discharging into the Colorado River. And so the Department of Energy took over this site, cleaned it up mostly, but also started a research operation there that I'll talk about later. So we have many sources of these contaminants in the environment. And the next thing I'm going to talk about is their geochemistry. And the key thing here is oxidation and reduction reactions. So these are elements that have multiple valences. And what that means is they can have multiple charges. All three of them can be in the +6 state. which means that the element itself is nominally in a +6 charge state. But then we can add electrons to them, or rather nature can add electrons to those elements. And that decreases their valence and completely changes their chemical properties and often changes their toxicity as well. So let's talk about this. So chromium can be in the +6 state, or we can add 3 electrons and change it to the +3 state. When it has the +6 state, it forms this chromium with 4 oxygens around it. Sometimes there's a hydrogen added to it, but that whole grouping is mobile in groundwater, and it's toxic because of its oxidizing character. And because it's mobile, we have groundwater plumes where in some cases the chromium just goes for miles. If the chromium accepts 3 electrons, it becomes chromium 3. See how I'm using these Roman numerals to indicate the valence of the element. So when we change chromium from the hexavalent form to the trivalent form, it becomes quite insoluble. So it precipitates out or adsorbs to surfaces. You want to consider it basically immobile. It also isn't nearly as toxic. Selenium has similar behavior. So it too has a +6 state, and it would be present as selenate in that +6 valence. Its chemistry is complex. It can also be in the +4 state, which is also quite soluble, somewhat less mobile. But then in the reduced state, when we add 6 or 8 electrons to that selenate, then, and again, this can be done by natural reactions or biochemical reactions, then the selenium could precipitate as elemental selenium, or it could form solids with metals. And all of these are insoluble. So once again, we take the oxidized form, which is very mobile and can be taken into, it can be taken in via drinking water or other sources, and is quite toxic. And we convert by reduction, convert to an immobile form. So that means that the contaminant is locked down and will no longer move. Same thing for uranium. Once again, we have a +6 state. It forms this odd ion UO2+, but then that tends to combine with carbonate in the water to form complexes which are quite soluble and mobile. But then under the reduced conditions, if we add 2 electrons to uranium, it forms uranium dioxide, which precipitates out readily and locks down the uranium. So all 3 elements, the thing you want to remember is oxidized means mobile and high environmental impact, and reduced means immobile and low environmental impact. So you can see where I'm going with this. When we look at a natural system, sometimes the contaminant plume is moving along, and just by natural reactions, it could be microbial reactions or perhaps abiotic reactions. Sometimes the subsurface is a reducing environment. It supplies the electrons to convert these elements to their immobile forms, and the plume just stops. It's possible for the plume to be reoxidized. I'll talk about that later. But this is our general story where we would like to know, is reduction happening? This reduction could be natural, or it could be actually artificially induced. Sometimes we'll inject reductants, electron donors into the subsurface and try to convert the contaminant to immobilize it. So a little audience participation here. So this map that you're looking at, this field site happens to be in Berkeley, California. And this is the chromium plume that existed there in the groundwater some years ago, decades ago now. And these contour lines are telling you that inside this highly colored area here, the concentrations were above 2 milligrams per liter at the time. Inside this line here, greater than 1 milligram per liter. And then in this less colored area here, greater than 0.5. And then finally, the uncolored areas are all less than 0.5 milligrams per liter. So quick look at this plume. You can see that they removed the source and the groundwater is flowing from top to bottom. And so this is what we call a beheaded plume. The source is no longer there and the groundwater is cleaning itself up in the upper reaches. But the core of the plume here still has concentrations in their highest or about at the time were about 13 milligrams per liter. And the question that we have is this plume going to continue to migrate past underneath the railroad tracks and out to San Francisco Bay? So if you look at the data here, so the black points are the measurement points and the contours are telling you the concentration. You have two hypotheses here. One is that this end of the plume down here, that there is no reduction happening. And therefore, that's just as far as the plume has gone. So really the shape of the plume could be due not to any reduction that's happening, or it could be strictly due to the contaminant history. The other option is that as we go from the core here down gradient, that decrease in concentration is mostly being caused by reduction. Now, if you're only using concentration data, can you tell the difference between those two hypotheses? You really have no ability. You just know what the concentrations are. Now, this is a snapshot. If you are willing to measure the concentrations 1 year and then come back 10 years later and measure them again, then you could probably infer if there is a great decrease in mass, you could infer that reduction is happening. But even then, there are other processes such as adsorption that might decrease the concentration temporarily, but that adsorbed chromium-6, which is basically sticking to the outside of solid particles, it's easily remobilizable, that's common. So, so it would be really nice if we had another way to get at the question, is reduction happening here? The concentrations, they're complicated. The concentrations are responding to advection, which is the movement of the contaminant with the water. It's also responding to dispersion, and that is the spreading of the contaminant plume as it moves with the water. There's adsorption, where the chromium-6 will stick to solid particles. There's dilution, which is essentially the mixing of clean water in from the sides. And then it even gets worse than that, because each well, the different wells are probably completed. They're probably drawing water from different depths. Some of them may have a wide drawing interval where they draw water in, and others may be narrower. And so groundwater systems, they seem simple. This diagram looks simple, but actually it's really hard to figure out if these reactions are happening. We really wish we had something that would get right at the problem and answer the question, do we have reduction here or not? And that brings us to the stable isotope ratios. So when I first got started in this business, we didn't measure chromium or selenium or uranium isotopes, but we had some clues from these lighter elements that had been studied already for decades. And we knew that those elements had, there were changes in these ratios. In each case, you'll notice that there is a heavier isotope compared to a lighter isotope. So essentially, we're counting up the number of carbon 13s, comparing it to carbon 12s. We do the same for nitrogen 15 and 14. We can do that for oxygen 18, 16, and sulfur 34, 32. So again, these have been studied for decades. And we knew that they had certain properties, and the quest for us was to see if that could work with heavier elements. And I have to tell you, I had pretty well-known isotope geochemists at the time who work on this kind of stuff say, oh no, those elements are too heavy, you won't see anything. Well, we have developed this, and it has worked. One thing that I'll mention is the uranium isotopes actually technically aren't stable, but the half-life of the 235 is almost 1 billion years. So for us working on decadal timescales, we don't have to worry about that. And it turns out, actually, even if you want to work on 3 billion-year-old samples, if an isotopic shift happened 3 billion years ago, we still have it today, even with the radioactive decay. So, we can treat the uranium isotopes as if stable. So why would you get changes in these ratios? What's going on is these are chemical reactions, these redox reactions, these reduction reactions that I'm focusing on. And the chemical properties of the lighter isotopes are just slightly different from the chemical properties of the heavier isotopes. one way that you can think about it is the lighter isotopes, they're a little more flighty, they're a little bit more, they're a little bit more energetic. In quantum terms, they have higher vibrational frequencies, they have higher 0-point energies, if you know what those things are. But again, you just want to think of the lighter isotopes as being a little more able to, for example, break a bond. So if you have a bond that has heavier isotopes on the ends of the bond, and compare that to a molecule that has lighter isotopes on the end of the bond, you can break the bonds bearing the lighter isotopes more easily. And so again, we knew when we got into this, with decades of work on sulfate, CO2, and nitrate, we knew that there is this general tendency for the breakage of bonds to favor the lighter isotopes. So we went into this. Oh, and so let me show you an example. Now, nitrate, imagine this is now, instead of a chromium plume, it's a nitrate plume coming out of some contamination source here, and then the nitrate is moving in this direction. Well, for a long time, we've known that if we measure the nitrogen 15 relative to the nitrogen 14, so again, how many nitrogen 15 atoms are there compared to nitrogen 14 atoms? The nitrate will become enriched in the nitrogen 15 as that nitrogen moves along, and some of it is removed by reduction reactions. That is because the reduction reactions are destroying the nitrate a little bit more quickly if it's a nitrogen 14 bearing molecule compared to the nitrogen 15 bearing molecule. So this is a classic, what we call a kinetic isotope effect. The lighter isotopes react faster, and that means that the remaining contaminant that has not yet reacted is going to be losing faster isotopes faster, lighter isotopes faster, and will become enriched in the heavier isotopes as it goes on. Okay, so then the game that we play is we measure the nitrate, we measure the nitrogen isotopes in the nitrate of this plume. If the N15 over N14 ratio is increasing with distance, we have good evidence that the reduction reaction is happening. And again, we knew this when we got into this work. The real question was, is this going to work for chromium and selenium, and then eventually uranium? A lot of what we have done over the years is develop measurement methods. And as Catherine mentioned, the measurements in particular for selenium isotopes, they're a nightmare, but let's not go there. So let's concentrate on the really good aspects of this. The practical knowledge that you do need to take away from this is the measurements need to be made on specialized instruments. These are $1 million instruments. It also is very time consuming to do the sample prep. And currently we do it quite manually. It could be scaled up as time goes on, and I think that will happen. But currently, minimum cost is $100 to $200 per sample. But I'm optimistic that in the future it will get easier. Okay, so my group set out to answer this question. Can we do the same trick with chromium, selenium, and uranium that people had been doing with other stable isotopes for a long period of time? And so we did a series of laboratory experiments, starting with a postdoc that worked with me, Mitch Herbal, and then moving on to PhD student Andre Ellis, master's student Jackie Kitchen, visiting scholar Jianming Zhu, and then Anirban Basu, who's here in the room, Emily Berna, Shanli Wang, Catherine Schilling, who's also in the room, Lisa Kirk, and Naomi Wasserman. All of these students and collaborators did experiments so that we could understand the workings of these elements and see if we had a chance to use them for environmental investigations. So now I have to digress for a moment and talk about the notation that we use. And again, what we're measuring here is we have very high precision measurements of a ratio. In other words, the number of chromium 53s compared to the number of chromium 52s. So it's always the heavy isotope, the one that has the greater number, that has more neutrons in it, and therefore it has that slightly different chemical behavior, and compared to a lighter isotope. the notation that we use is this delta, which is a parts per thousand shift from some standard that all the different laboratories use. And so we could have used percent. We could have said that, well, this sample is 1% greater than the interlaboratory standard. But because the changes are so small, we chose to use per mil. So, but basically all you need to remember is that a positive value of delta means that there is an enrichment in the heavier isotopes in whatever we're measuring. Negative means it's enriched in the lighter isotopes. Then I'm also going to use this epsilon. So I've already talked about how there is a preference for the lighter isotopes to react faster. And you would ask then, well, how much? Is it big enough to measure? And we need to be able to calibrate the changes that we see in nature. And so we've done laboratory experiments to measure epsilon, which is really just telling us how different is the product that's being produced at an instant in time compared to whatever is reacting. Okay. So epsilon is really our calibration factor. And the way to think about that is we know the mathematics of this situation. If we have a simple situation in nature, and reality is a little more complex, but to first order, if we want to know the amount of chromium-6 in this case that is being removed by reduction, we know what the mathematics look like, and we know how much delta should increase as the amount reduced, i.e. immobilized, increases. We know that it will follow a curve like this. And this is a curve for an epsilon of 4. If epsilon were a larger number, we would simply have a steeper curve of the same shape. If epsilon were smaller, we would just have a shallower curve of the same shape. So you can think of epsilon as our calibration factor in our mapping of how much reduction has occurred to the isotope ratio. I'm going to make a very long story short. We did many laboratory studies, and there were some other laboratories around the world that worked on similar problems. The reaction going from chromium-6 to chromium-3 changes the chromium-53 to 52 ratio by about 1.8 to 4.3 per mil. The values are negative, and that's just telling you that the lighter isotopes are reacting faster. You may be a little bit worried that this is a pretty wide range, and this was our first complication that came at us. We realized that there wouldn't be a single number. Kinetics, anybody who's had a PChem course or done any kind of chemical kinetics, you know that it's horrendously complex. And so with the isotopes, we do have some complexity based on the kinetics, the reaction mechanism, and sometimes the conditions of the reaction. We can get a higher epsilon or a lower epsilon, and that's just making life a little bit more complicated. But the good thing is, for all the reactions that we really care about, we found that the isotopic fractionation occurred. For selenium, again, we found here a wider range, but again, we're always seeing these shifts in the isotope ratios, both for the reduction of selenium-6, selenate, to selenium-4, but then also selenium-4 going to the elemental selenium. That is the immobilized one. So we have these large isotopic fractionations here. Our precision for most of these measurements is about 0.1 per mil. So if you think about that, that's 0.01%. And so those of you who work hard in laboratories, you know that just getting 1% precision for most measurements is doing really well. Well, this is why our measurements are so difficult. We have to be extremely precise. So
can I briefly
yes.
ask, the range for the uranium, is that a little bit confusing, the fact
Yes.
that you go in both directions?
Oh, thank you. Thank you. Thank you for stopping me, because the last thing I wanted to say about this slide is that uranium is strange, and we see a similar isotope effect, but it is backwards, and it's backwards for reasons that I don't have time to talk about today. I'm looking at the time. And there is a strange interaction between electrons and the nucleus that causes the kinetic isotope effect for uranium to go backward. And then there is debate in the community as to whether that is always true or not. We tend to think it is always that way, but there's more work to be done. And this is the slide where I could talk about the crazy theory behind this. And I'm just going to move on. So let's get into a field study. And I'm going to talk about chromium isotopes. I realize probably most people in the room would be more interested in selenium and uranium. The thing with chromium is that we've done the most work on chromium. We have the best developed studies. And this field study here, I've already showed you the concentration data. Let me show you the isotope data. This is really nice. So in this system, I showed you the map view earlier. Here is our contaminant source shown here schematically, and the chromium moves through this system. In most of the system, there are no reductants that are changing the valence of the chromium-6, but there are these sedimentary layers that are finer grained and contain organic matter and probably ferrous iron that can act as electron donors and change the valence of the chromate. So go back to, here's where we were a few minutes ago, and you look at the concentration data, and you really want to know, is this reduction that is happening as we go down from the core of the plume, down gradient, down the groundwater flow direction to here where the concentration is approaching 0. Let's see what the isotopes say. Well, you tell me what the isotopes say. These are numbers that are, again, giving you the per mil deviation of the chromium 53/52 ratio compared to a standard. And as you look toward the top of the diagram, closer to the source, those are the groundwaters where the contaminant has been in the ground a shorter period of time. And we see that the numbers are lower. And then as you march downward, farther along the groundwater flow direction, the delta values are getting higher. That's telling you that the chromium 53 relative to 52 is increasing. That means that the groundwater chromium 6, this is what we're measuring, chromium 6 only, is enriched in the heavier isotopes. Where did the lighter isotopes go? They were taken up by reduction, or they were taken up a little bit faster by reduction. And so this value down here, down by the railroad tracks, that was an interesting sample to take with the Amtrak going by at 70 miles per hour. But the value down there is fairly high. And that's a strong indicator that reduction happens. One thing that I didn't mention earlier is at the time we had looked at some other processes that could be happening, such as absorption in this system. And it's really only reduction that causes the shift in the isotopes. There's a reason for that. These redox reactions greatly change the bonding of the chromium, and that is what changes the isotope ratio. Okay, so the isotope data qualitatively tell us that there is reduction happening. Can we do better than that? Can we be quantitative about it? So just to summarize, the delta value is increasing with increasing distance. We infer chromium 6 reduction, and that means that we have natural attenuation of the contaminant. In other words, it is reducing, it is being precipitated out as chromium 3. It will never make it to San Francisco Bay. And the amounts of chromium 3 that are being deposited in the aquifer are actually not that great compared to the natural inventory. So this is a big success story. And this is without any artificial intervention. This is just the naturally occurring reducing sedimentary layers in this system. Okay, so now we'd like to answer the question, can we get quantitative about this? Can we tell you, is the chromium 6 30% reduced, 90% reduced? How far are we going here? Well, this plot here is a little bit complex. On the x-axis, we have the base 10 log of the chromium 6 concentration. On the y-axis, we have the delta value. So the way you read this diagram is we start out with HPO9, that is the highest concentration well in the whole array. And it is right in the central axis of the plume. And then as we go down the plume, we're following along the filled symbols here. And we see that with increasing distance, the isotopes are increasing, but you knew that already from the map. Here is a line that tells you what would happen based on an epsilon value of 2.74 that we obtained by laboratory measurements. So we took some of that green sediment, some of those green sediment layers from the site, we incubated them in the laboratory, and we found the epsilon value, the size of the isotopic fractionation for the reactions that were happening in that material. And we inferred that the entire site was behaving the same way. And so we were a little bit disconcerted at first because the water should be following this line, and yet they're going at a much shallower angle upward. So we thought about this for a while, and based on some other work that we were doing with selenium isotopes in a different system, we realized that we had a problem, or that we had a challenge here related to heterogeneity in the system. So I'm going to go through this very quickly because I have to move on quickly. But remember, the system has zones where the chromium-6 can freely flow without reacting. And the chromium only reacts inside these limited zones that have the organic matter and possibly the microbes and the iron too, the divalent iron, the ferrous iron. So under those circumstances, we actually predict that there is what we call a reservoir effect. So some of you, if you have dealt with these sorts of things with geoscience and isotopes, this is for you. Basically, because there is heterogeneity, there are zones where the chromium is simply moving and the reaction is only occurring in these limited zones that have limited flow of the water through them because they're finer grained. We expect to see a diminishment of the size of the isotope effect by at least a factor of 2. And I don't have the time to go into why that occurs, but it's a well-known phenomenon known as a reservoir effect in the isotopes. So the dashed line on this diagram is what we would infer, what we would guess if we have a kind of standard reservoir effect. We've created a simple model. And that is not too far. So this trajectory here is not too far from the actual data. So we think that dilution, which is going to move you directly left, is happening to a small extent, and that can move you from the theoretical line to the actual measurements out here. And so the conclusion is actually, it seems like the concentration data and the isotopes tell the same story. In other words, the two are fully consistent for this site. There are some assumptions that go into this, but coming out of this study, we were highly confident that chromium isotopes work in the real world as an indicator of this reduction reaction, which immobilizes the chromium and is a really good thing to happen, either through natural causes, as happened in this site, or through artificial injection of reductants. So then the answer, if you were wondering how much reduction has happened when it gets to the railroad tracks, 95% is our estimate from our analysis and our model. So again, we think that the chromium isotopes are very effective. Let's move on to uranium then. Can we use uranium in the same way? So now we're back to the Rifle, Colorado site here. And these arrays of red dots that you see, these are the different plots of experiments that were done at the site. The groundwater flow is generally, let's see, I'm not remembering exactly. I think it's coming across in this direction. And again, some of that uranium is discharging into the Colorado River. Plot C right here, actually, there are no red dots for plot C because I want to show you them here in broader view so that you can see the details. So in this diagram, this is distance in length. In this, on the x-axis, and so this is the flow direction is from left to right. And across the flow direction, you can see we've got about 12 meters across. And then in the down gradient direction, the downflow direction, this is about 18 meters long. This was an experiment where the uranium concentrations that you'll see in a moment were fairly high coming in from the left. And those were measured in these upgradient wells. Then this pink zone here is a gallery of wells where they injected acetate into the subsurface. The acetate is food for the naturally occurring microbes at this site. So without any other manipulations, simply injecting electron donors that the microbes could use allowed those microbes to reduce the uranium and immobilize it, take it out of the water. We are going to look at both the uranium concentrations and the uranium isotopes in this one down gradient well. It's only 2 meters or so down gradient of the injection, so it responds quite quickly to the microbial growth. And as you'll see, the uranium concentrations go very low quite quickly. So I think, does anyone have any questions about the makeup of this experiment before I move on? The details are pretty important. I think I've hit them all. Okay, so let's go look at the data. On the x-axis of this plot, we have time. So this is starting on July 1st of 1 year and going to July 1st of the second year. And then here's almost July 1st of the next year after that. These pink zones are once again showing you, in this case, when the acetate injections happened. So in year one, toward the end of the summer, they injected acetate for about a month. And the uranium concentrations started at almost 200 micrograms per liter. And then they dropped dramatically as the microbes got busy because they had been fed the acetate. Then shortly after the acetate injection stopped, the uranium concentrations, the uranium re-entered, moving with the groundwater, and the concentrations went back up. The concentrations even went a little bit higher than they started. There are a few reasons why that could have happened. One could be remobilization, another could just be higher concentrations in the water coming in. In year two, you can see that they hit the aquifer with acetate longer, and they injected a higher concentration. The goal there was to see if they could lock down the uranium for a longer period of time so that it wouldn't bounce back up immediately. And they succeeded in that. And the uranium concentration stayed low for a while, but eventually rebounded back up close to where they started. Now, let's go to the isotopes. The isotopes are the green boxes, and just a glance at the isotope values tell you that the uranium isotopes are responding quite strongly by about 1 per mil, more than 1 per mil, which for uranium is fairly large. The laboratory experiments tell us that the epsilon value is up to 1 per mil. And so that's the rough scale of the shift in the uranium isotopes. And then as the uranium concentration rebounds, the uranium isotopes go back. And again, that's because fresh groundwater is flowing into the plot after the acetate injection has shut off. Then the isotopes stay at their initial values, or they're back at their initial values for the rest of the year. And then when they get hit the second year, they go negative. And then something strange happened at this point in time where they turn off the acetate, and actually the isotopes go deeper into negative territory and then start to rebound. We have spent a lot of time understanding this somewhat complicated behavior, and I don't have time to talk about this second phase here, but in general, the uranium isotopes are tracking the reduction of the uranium-6 quite effectively. So let's see, and again, I'll stop. Are there any immediate questions? All right, so the basic takeaway message is, like the chromium isotopes, the uranium isotopes seem to be working pretty well. We can put a very simplified model on this, known as a Rayleigh distillation model. Essentially, this is the same as the curve that I showed you before, with the chromium isotope going up as a function of the amount of reduction that has occurred for uranium. In the early parts of the experiments, we started here, and the concentrations go down, meaning that moves us to the left. So the x-axis is concentrations, isotopes are on the y-axis, and the concentrations and isotopes are following a pathway that is consistent with epsilon equal 0.85 per mil, and that's what we get from our laboratory experiments that Anirban did so well. Then in the middle of the year, the incoming concentration decreased. We know that from the upgradient wells, so we have to shift our model in this direction. And we did some modeling to take out some hysteresis effects, some memory in the aquifer, and we find that the data fit quite well when we do that modeling. These are the isotope values and the concentrations plotting with each other. So again, it really seems like the uranium isotopes are working quite well. Now, one other question that we had is, you noticed that at the end of the first year, the uranium isotopes and the uranium concentrations rebounded. So the kind of scary question is okay, we've done great bioremediation, we've immobilized some uranium, but is it then oxidizing back out immediately? So we did some work with that. This is Noah Jemison's work. And just to get quickly through it, they injected into the same plot, they injected some nitrate as an oxidant. And let's see, the concentrations are here. Here is days after the start of the injection. And the top here, we put in a special tracer that would tell us when the oxidant was coming through. So as the oxidant comes through, we see the uranium concentrations, the filled circles increase quite a bit from 200 micrograms per liter all the way up to 600. So we're mobilizing a lot of uranium by oxidation, and the isotopes are tracking that quite well. So because when we were doing the reduction, we were producing solid uranium that was enriched in the, let me make sure I get it correct, enriched in the heavier isotopes. When we oxidize it back out, we see that signature coming back out. So again, a success story. We think these isotopes are working quite well. I'm going to skip this plot in the interest of time so that I can give you just one very quick example of how selenium isotopes seem to be working the same way. This is an open pit coal mine in a beautiful area of just on the western slope of the Canadian Rockies in what's known as the Elk Valley in British Columbia. These coal mines are producing a lot of selenium that's getting into the water there. The coal company is working extremely hard to decrease their discharges of selenium. And one of the things that they do is, you can see these waste rock piles here. One of the things that they have been doing is creating areas where they have these subsurface bioreactors, where they have a large zone where they pump water in one end, they pump it out the other end, they add acetate or some other electron donor, and they're hoping to reduce and immobilize the selenium. So we've been helping them out by doing some selenium isotope measurements, and just very quickly, the injected water in one of these systems was up at, I don't have any units on this, but it's micrograms per liter. So up above 100 micrograms per liter in the injected water. Their treatment system is very effective. It is moving the concentration much lower to the left side of the diagram. So, as you can see, the data points, the concentrations are getting much lower. And here is the isotopes on the y-axis. These are some of the highest values we've seen anywhere for selenium isotopes. because the reduction is so strong, it's almost completely taking the selenate out of the water, and the isotopes are going up, 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 up. For some of the samples, some of the other samples, there's something more complicated going on, and some of them don't seem to be, it seems like it's just dilution in this system. So these, again, we think that this is telling us that the selenium isotopes work in the real world, not just in the laboratory. I have other examples for selenium isotopes, but in the interest of time, I just want to touch on one other thing and then I will stop. A lot of times people ask us if we can trace different sources. Can we fingerprint different sources of contamination using these stable isotope tracers? I'll just make a long story short and say that we have two worries when we try to do that. Sometimes we go into these systems and we find that the different sources of contamination just aren't different. They don't have different fingerprints. They don't have different signatures. Sometimes we see that they have different signatures, but in that system, perhaps we have reduction happening. And so the signatures are being overwritten by the geochemical reactions. So I want to suggest caution, but I want to suggest that people try. So the way to try to succeed with tracing stable isotopes is to in your system carefully try to distinguish the different sources. If you don't have a way to get pure samples of the different sources that you have in your system, then that's going to make it more difficult. And then also be cognizant, if you are measuring samples in a system, trying to find out which source is dominant in the system or where the different sources of contamination are flowing. Be aware that there could be reduction, there could be other processes that are changing the isotopes along the way. So that's all I'm going to say about that. And I just want to end to summarize what I talked about related to the redox reactions. So chromium, selenium, and uranium all exhibit these isotopic fractionations, these isotope ratio shifts that we think are very good diagnostics of reduction in these groundwater systems. And this reduction is critically important for understanding the mobility of the contaminants. So we also think that in some cases, remobilization is something that we can detect. It's more complicated than reduction, but we think that it can be useful. Now I want to back way off to the 10,000 foot level and talking to a crowd that has many different people coming from many different directions. I want to review some of what I had said and think about it in terms of biomedical, whatever. So here in the groundwater systems, when we look at concentrations, which are our standard tools that everybody's been using forever, we see that the concentrations are affected by many processes. And so often it's impossible or difficult to disentangle one process from another. The isotopes can be targeted very strongly at one or maybe a few different processes which change the isotopes. Right? They're often much simpler than the concentrations. The other thing I'd like to say about that is that they're really coming at these questions that we have from a different direction. So they're not just giving us redundant information that the concentrations are holding, it's fundamentally different information. And even though, as you saw with our chromium isotope study in the field, there are complications. It's the natural world is always very complicated, just like the human body is quite complicated. We expect those complications to come up, but the isotopes will always be somewhat orthogonal and give us information coming from a different direction. Last thought is I'm just going to show you the periodic table, and there are many elements included here. I talked about 3 of them, and there are many others that are contaminants that are being studied. And with that, I'll return to our group from some years ago with Urbana and Catherine in it. And thank you. Sorry for going a little bit long, but I'd love to take questions. Get in the back.
I have one like, after the acetate injection, how much of it is because like the organism that actually benefited from the acetate as like a food source, ended up dying already, so like, in the process of decay, then you lose all your oxidizers, and so you have like a reduced, a reducing environment. And would that be contributing to the drop? And then once the biofilm recover, then, it becomes oxidizing again, and so you see the release of uranium back into the water.
So there's a few different questions there. There are some people that are quite famous for studying these naturally occurring microbes, and they know that there is direct reduction of the uranium-6 in the laboratory. We infer that it is done, that's the major mechanism in the wild. The abiotic reactions are possible, but they're probably slower, so we think it's probably microbial. The biomass itself, Anirban is probably a better expert to ask on this. After the bugs die, we like to call the natural microbes, the bugs.
Yeah.
After the bugs die, that biomass, I don't know if it would have, I think for uranium, you have to have a pretty strong reductant. I think you really need to do the reduction enzymatically. Just generic biomass probably wouldn't have enough reducing power. But again, maybe Anirban can comment on that.
So
or maybe we don't know.
This particular experiment, they looked at the 15S RNA sequence from the samples, from the water sample.
Speak up for the people on
And
Zoom.
they found species like geobacter, the iron reducing bacteria, they were active in the aquifer. So that was the one of the evidence that the particular reducing bacteria grew. It is possible that they also reduce iron 3 to iron 2, so a little bit reduction by iron 2, but that process is what we know so far a bit inefficient in absence of surfaces. So in the solution, it's more like the enzymatic reaction that is the more efficient reduction process.
There are a lot of cytochromes that can reduce uranium directly.
Yeah.
Yeah, but iron is an inefficient uranium reactor.
And I'll have a special specific question later.
But your question is right on. We wish that we could use the isotopes to diagnose different reaction mechanisms. We think that might be possible in some settings. In the selenium case that I was just talking about, I only showed you the early data. We're now scratching our heads because we have some later data from that same system where we think that the reduction is continuing on quite strong and the isotopic fractionation went way down. We think we have some hypotheses to test about what's going on there. Other questions?
So, I have many questions, but I'll start with the reversibility of your uranium-4 oxidation.
Mhm.
It's somewhat surprising given that a whole range of people were hoping that it would be less reversible. But the time scale of that reversibility is pretty short, it looks like from your experiments.
Well, most of the rebound in the uranium concentration was simply contaminated water re-entering. It looked like the rebound went a little higher than the initial concentration, but in that site, the initial concentration, incoming concentration is always bouncing up and down. So there's really no way to be sure that there was oxidation happening immediately. Now, there is evidence that there wasn't very fast oxidation happening because the oxidation experiment that I showed you was from the same plot years later, and there's still a lot of uranium down in there. And we just hit it with some nitrate and it comes out. So and again, Anirban, you might know a little bit better, but my thought is that it's probably starting to trickle out, that it's probably starting to oxidize very slowly. The dissolved oxygen is not a very good oxidant. In fact, they sparged the groundwater with air, and when they didn't use nitrate, but they were trying to use air as the oxidant, very little happened. Then they hit it with nitrate, and boom. Probably because the microbes were busy again, making some nitrite. So, so I think it's a little bit reversible, but I think mostly it's the uranium is pretty well locked down.
People can feel free to leave if you have to go, because I know we are over, but then.
Yeah, so I'm just curious about like how do you think of the role of soil and matter and like these clay minerals and how they are affecting this reaction or oxidation reactions like for these organic matters, they many of them can like these bending rings have these large pi systems. They could hold these electrons. Maybe they can, I don't know, like find the reduction, like dynamics or like kinetic. So it's kind of what do you think of this? Directions.
I think that there are many systems, for example, in the Chicago area, that the natural organic matter is just perfectly good. As natural reductant that the microbes feed on. We know that chromium plumes in the Chicago area, they just don't go anywhere. We don't need to do isotopes. The chromium plumes just stop immediately. So natural organic matter can be a perfectly good reductant or a perfectly good food for the microbes either way. That's for chromium. For selenium and uranium, it requires a little more reducing power to get them. But I think, again, in many systems, the organic matter is already there. But then there are other systems that are highly oxidized, that are very organic rich, or sorry, they're very organic poor, very poor in organic compounds in the natural soils and subsurface materials. So those are the ones where in some cases we have doubted that there's any possibility to have reduction. And then we look at the isotopes and sometimes we see a little bit of reduction. So I think you're right on that the natural organic matter you look at that and often there's plenty of fuel there for these reduction reactions. And if it's not going fast enough, then we can inject.
Can I ask my other question?
Go for it.
No, I'm saying, I was hoping that some people who work in the isotope world would have like a solution to one of our challenges. So I work mostly with organic contaminants. And what we've noticed sometimes is that there would be a mix. So to be concrete, we would somehow work with polychlorinated or polybrominated compounds. But then there are natural sources of polyhalogenated compounds, but then we know that there are also the anthropogenic ones like your PBDs and your PCBs. But when we detect like an unknown polyhalogenated compound, is there a way to use chlorine or bromine isotopes in this specific case, differentiate or determine whether they're anthropogenic or natural?
Probably. We already know
That makes me happy, but.
for industrially produced, I don't remember all of the different compounds, but certainly TCE was done. Well, yeah, maybe 20 years ago. And they looked at different batches from different manufacturers and looking at the carbon isotopes and the chlorine isotopes, which both can be done, not by our group, but by other people. The guy to talk to is Neil Sturcio at Delaware. So they found that they kind of made a map for the different sources and their isotopes were quite different. But then again, you have to be careful in the system. You may have reduction, dechlorination reduction reactions that would change those isotopes. So careful, but it's I would be optimistic.
Yeah, and it's tricky when you have poly ones, because you're seeing like a mix of different numbers of bromines and chlorines, and then you have the 35 and the 37 in different proportions, and then like how do you calculate how much is 35 or 37 and then make the ratios and, yeah, I don't know the answer, so.
I think you can just do a bulk measurement, you know, just mix up, take whatever is in the compound, measure all the chlorine. And even though there are different sites, just put it all together, and that would be your starting point. I would guess that you will find that the natural sources and the industrial, the anthropogenic stuff would be different.
Thank you.
Yeah.
Wonderful. Thank you so much.
Thank you, everybody.
We can continue the conversation.